this video, I'll be discussing throat cancer in a species of dragons that may be caused by tumor suppressor or proto-oncogene. Tumor suppressor genes prevent bad cells from dividing when they are not supposed to and prevents uncontrolled growth. It results in a tumor if no normal allele is present, but it must disrupt both copies of the gene to lose cell cycle regulation. This is a recessive mutation. A tumor suppressor gene is the equivalent to a brake pedal in your car. In driver's ed, it's possible to have two brakes, one for you and one for the instructor. If you are unable to brake, the instructor could do it for you. Same thing here. You need to disrupt two copies in order to lose the cell cycle regulation. Proto-oncogenes prevent bad cells from dividing and promote cell division. A mutation in a proto-oncogene can result in an oncogene that allows uncontrolled cell division. You need a mutation in one copy of the proto-oncogene to get a tumor. This is a dominant mutation. The proto-oncogene acts as an accelerator. It keeps the car in motion or promotes cell division. Typically, you only see one accelerator, which can be directly correlated to only needing a mutation in one copy of the proto-oncogene. A tumor is a distinct mass of abnormal cells that do not have normal controls on cell division. In a benign tumor, the cells remain localized and do not invade surrounding tissues. But in a malignant tumor, the cells invade surrounding tissues. Once you get to the metastatic tumor, the cancer cells spread and establish secondary tumors in other sites of the body. 3 genes X, Y, and Z contribute to throat cancers in dragons. In benign tumors, there tend to be mutations in both alleles of gene X, while genes Y and Z have wild-type alleles. We can see both alleles are disrupted in gene X. This means that we have a recessive mutation, and we can say that gene X is a tumor suppressor gene. In malignant tumors, there tends to be mutations in both alleles of gene X, which we already figured out was a tumor suppressor gene. But now there's one mutation in gene Y, and Z still has no mutations. Since there's one mutation in gene Y that leads to a malignant tumor, we can say that we only need a mutation in one copy of gene Y to get a malignant tumor. This is the dominant mutation, meaning that we can say that gene Y is a proto-oncogene. In metastatic tumors, we still have the same setup as the malignant tumor, but now we have a mutation in one copy of an allele in gene Z. This also means that this is the dominant mutation, and we can say that gene Z is a proto-oncogene. Normally in the X gene, you would be able to use your brake when approaching a red light or be able to inhibit cell division when needed. This is disrupted. This is the equivalent of your car's brake going out, and nothing is slowing the car down at a red light or inhibiting cell division. Disrupting the function of the tumor suppressor gene causes the organism to lose regulation of cell division. This cell will continue to divide even when it's not supposed to. This will eventually lead to cancer. Normally in our X and Y proto-oncogenes, you would be able to accelerate freely, but be able to stop when approaching a red light or a stop sign. Also, you would be able to regulate cell division, but stopping when bad cells are present. When a proto-oncogene is disrupted, this is the equivalent of your car constantly accelerating without being able to pay attention to the traffic lights or the stop sign. Disrupting proto-oncogenes will cause the cell to promote cell division without ensuring that bad cells are prevented from dividing. This will cause the cell to go through many cellular divisions while accumulating several mutations along the way. This will also lead to cancer.